Everyone, and welcome to this episode of Foundry Fireside. My name is Lori from the Foundry Communications team, and today I will be chatting with one of the Foundry's postdocs, Alex Lin. Alex works with Peter Ursius at the Foundry's National Center for Electron Microscopy, and his research is focused on using electron microscopy to study materials made by living things. Welcome, Alex. All right, Alex, how about we get started with you telling us a little bit about your project?
My project involves trying to understand the structure and properties of biomaterials. And these materials include hard ceramics, bone-like structures, and carbonates, such as seashells. And I have been working with a team from the Joint Genome Institute that have been engineering microbes to produce these kinds of biomaterials. And my role specifically involves understanding these structures using the transmission electron microscope, the TEM. By understanding the nanostructures of these materials, we can then determine which applications these materials can then be used for. More recently, I have been focused on looking at the different pathways that lead to these minerals, and I have been working with the biofacility at the Foundry to actually understand how these cells uptake ions from their environment to produce these inorganic structures.
That's really cool. Now, the Foundry has two of the most powerful electron microscopes in the world in the TEM microscope. How are you using them to study these biomaterials?
So in the TEM, we can use them to take images of the nanostructure of these materials. And also we can use X-ray spectroscopy inside the TEMs to understand the chemical compositions of these materials, which is actually really important information if we want to engineer these materials for specific applications. Additionally, the TEM-05 currently has an electronically controlled stage, which then we can write computer programs to operate these microscopes autonomously and remotely over long periods of time, which opens up the doors to perform high throughput transmission electron microscopy. So this actually helps us collect large amounts of data, which then give us good statistical representation of different nanostructures across our entire sample. So some of the most recent advances in that technique allows us to actually collect hundreds to thousands of images per sample. And then we can actually record thousands of observations of the same kind of structures over time.
That's awesome. So can you tell me what makes you excited about this project?
So this project is exciting because there's a lot of different potential avenues for us to go from. So currently we have engineered yeast and bacteria to produce different types of hard inorganic materials. However, these materials can then be used for other critical applications such as infrastructure and construction. So it's exciting because a lot of our current challenges with these industries is that they require large amounts of energy and carbon. So using microbial synthesis to make biomaterials and also just materials in general actually opens up the doors to a lot of potential opportunities, which allows us to basically reduce our carbon footprint in the manufacturing sectors.
So when you say using microbes for infrastructure, does that mean one day we'll have self-healing concrete?
Yeah, absolutely. That's definitely a very active research area currently. So there's a lot of different research groups and small companies looking at this problem right now. And I think the really cool part about microbial synthesis is there's a lot of different pathways in each organism that you can tune. So for us right now, we're interested in understanding how these organisms uptake calcium from their environments. But you can also think about how these organisms can uptake other elements, and that opens up in a lot of opportunities to many, many different applications and possible materials that we can synthesize.
That is so cool. Okay, so electron microscopy and studying how microbes make biominerals seem to be really different fields. How did you get into this area of research?
Yeah, so actually that's a really interesting question. When I first started getting into electron microscopy, I actually did a summer internship at McMaster University in Canada. And for that project, I actually used TEMs to look at dental implants and how the bone heals around these implants over time. And that was a really fun project to be involved in because it was using a really interesting state-of-the-art tool to probe biological processes, but also involves material science, which I got my degrees in. So that kind of got me into projects that involve understanding biological mechanisms, but also that touch upon material science principles. And in graduate school, I actually did a PhD involving a lot of different TEM techniques. However, that project was mainly focused on material science. And then once I got to Berkeley, there was a really interesting opportunity to take part of this project, which is a collaboration with JGI, and our ultimate goal is to actually find a way to decarbonize the economy. So I thought that was a really, really interesting opportunity and got involved with that project then.
That's really great to see how your undergraduate experience informed what you're doing now. And all right, I have one last question for you. What is the big question or next step for you in this project?
So the big next step with our project is to understand how our yeast platform is transforming calcium ions into hydroxyapatite, which is a bone mineral that is found in all organisms, essentially. So the answer to these questions will actually help us understand how we as humans form bone. And that understanding can then be used to develop alternative therapies or other pharmaceutical products that can help treat bone disorders. And it's actually a really interesting project because this next step involves combining different modes of microscopy, such as TEMs, but also optical microscopy, such as fluorescent microscopy on the 5th floor in the bio-nano facility. So by using both modes of microscopy, we can then actually understand how the cells are consuming calcium from their environment and transforming that into inorganic structures. So that live process was previously almost impossible to get with TEMs alone. So by combining the different tools at the foundry, we are then able to actually understand the exact mechanisms that's happening inside the cells. And that mechanism can then be used to help us control and modulate how we want to design our materials.
Wow, that's all really cool stuff, Alex. Thank you so much for spending your time with me today. And for all of you watching, thanks for tuning in. And be sure to check back soon for more episodes of Foundry Fireside. Thank you.
Thanks, everyone.